are the important properties and functional properties that you should remember from each of these? First off, arteries are thick and they're elastic, right? Very thick, especially with that smooth muscle, and elastic, which means that they have low compliance. They also have low resistance. All right, don't confuse the arteries with the arterioles. Arterioles are where we have the high resistance. Arteries shouldn't have high resistance. Otherwise, blood would not leave the heart with enough force to make it all the way through the entire circulatory system. So it's really important that they have a thick elastic wall. They have low compliance. They can actually kind of hold force, hold tension, and sort of create more pressure, but they should have low resistance. For this reason, we call them the pressure reservoir, right? The pressure reservoir. Okay. And one of the things that we said in terms of comparing them to veins is that we'll see a large pressure change for a small change in volume. Large pressure change for a small change in volume. Okay. And they also have the ability to recoil, right? We talked about how they can capture force in their walls so that during both systole and diastole, the flow going out to the vasculature remains constant, even though the aortic or systolic valves would close, right? So we definitely want to remember that they have that recoil. And then we also want to think about our arterioles. Arterioles, here we're going to see high resistance. So high resistance. We also see the greatest drop off in pressure because of that resistance encountered. So the greatest drop off in pressure. And then in terms of their radius size, we talked about different factors, the nervous system, intrinsic and extrinsic, that could control the size of the radius. So let's talk about the radius size. Couple things here. First of all, we want to remember that the tone here is because of the smooth muscle that is found here. So the tone is due to smooth muscle state, right? The state of the smooth muscle, whether it's constricted, which is contraction, or whether it's relaxed, which causes vasodilation. So vasoconstriction is going to result in decreased radius and increased total peripheral resistance. Vasoconstriction means radius gets smaller, resistance increases. While vasodilation this means radius gets larger, right, as that smooth muscle relaxes, and then TPR goes down, resistance goes down. So definitely recall how the state of the smooth muscle influences the radius size and the resistance encountered. Now, as far as the factors that we described that basically regulate the size of the arterial radius, what are some of those factors? We divided those into intrinsic and extrinsic factors. So let's talk about intrinsic versus some of the extrinsic factors. All right, intrinsic, one was active hyperemia. And I won't go into the details of active hyperemia here, but if you're not remembering this, you're not recalling this, please go back and review this. But basically, increased metabolic demand to the tissue means that we're going to have more flow, relaxation of the smooth muscle in order to bring more oxygen in and in order to clear more of those metabolites, such as potassium, nitric oxide, um, or even CO2, right? So some of those gases as well. So go over this, but active hyperemia in response to increased metabolic demand. The second one was reactive hyperemia. Reactive hyperemia, and this was in response to an obstruction. So a previous obstruction that has now been cleared, now we're going to have this reactive response where we increase flow for that reason. So the reaction or the response is the exact same. The cause or the trigger is different for both of these, right? Reactive versus active. And then the third intrinsic sort of regulatory response was the myogenic feedback, right? The myogenic feedback, or what we call the stretch response. Basically, when you increase the flow, the smooth muscle responds by contracting or constricting down, which we said was a protection to the vessel, but also just to regulate the flow so that there's not too much or too little flow going to that area. All right. And then as far as extrinsic factors, this included things like the nervous system, hormones, neurotransmitters, if it's the neuromuscular junction, the neuroendocrine junction. So this basically the sympathetic nervous system. And in terms of that, we said a few different things here, that norepinephrine binds to alpha receptors and brings about vasoconstriction. I'm running out of space here. Vasoconstriction, all right? However, when we have epinephrine, epinephrine can bind to beta receptors as well as alpha receptors, but this is where we talk about that dose-dependent relationship. So if epinephrine is in high concentrations, it binds to both, okay? Epinephrine at high concentrations, I'm trying to figure out how to write this with this little space, but epinephrine at high concentrations, it binds both alpha and beta. Alpha is going to be widespread vasoconstriction everywhere except the brain. Beta is going to be just to cardiac and smooth muscle. But at high concentration, it binds both. Alternatively, at low concentration, it binds beta only. Okay, that is to say that at low concentrations, it's only causing vasodilation on cardiac muscle and smooth muscle. At high concentrations, it's going to cause vasoconstriction everywhere except the brain and vasodilation more specifically to cardiac and smooth muscle. All right. So definitely go over this. And then remember, this applies everywhere except for the brain. You see, we have a question. Oh, so the question was about the equations. Well, we have to use all the equations that we have and be able to plug those numbers in. So the ones that I highlighted, the mean arterial pressure, cardiac output, total peripheral resistance, that equation is important to remember, to be able to apply to questions that may ask about the relationship, as well as to be able to plug those values in. And again, it's going to be small values that you can pretty much do in your head or one paper, but if you do need to use a calculator, you can. And then same for the Starling's forces. You should be able to plug in your filtration force, your absorption forces. And again, it's going to be pretty, easy numbers to work with, even numbers, and fairly easy, addition, subtraction, okay? So yes, you should be able to apply those two that we highlighted. Okay, let's move on to capillaries. Capillaries, some of their important features, they have a thin wall. which makes exchange of materials very easy, very important for this part of the vasculature, so diffusion can happen here. Right? One cell layer thick, very thin. They also have the greatest total cross-sectional area. Okay. Let's talk about the three different types that we described. So we talked about continuous capillaries, and these are the most abundant, right? Most abundant. These have endothelial cells with very small pores. Small pores. However, big things like lipids, big things like proteins, those cannot get through these pores. Then we have fenestrated. And fenestrated are going to have large pores, okay? But they still have a continuous basement membrane, right? Remember we said that they still have these fairly continuous on the basement membrane, but larger pores to where bigger things can pass through, bigger proteins, bigger particles can pass through those large pores. Finally, we have discontinuous, also sinusoids, and these have very large gaps, right, in between, and then they also have a discontinuous basement membrane underneath. So even larger gaps, really big channels in between the cells of these discontinuous. So know your three types of capillaries. We also talked about examples in some instances. Continuous is the most abundant. Fenestrated, we talked about in parts of the kidneys, parts of your endocrine glands, like your thyroid, um, your pituitary, and then discontinuous, mainly the liver for discontinuous. Let's talk about the regulation to capillary flow. Right. So I described this earlier on the other slide, so I won't go into the details here, but again, know the structure of these, know where they're found, meta-arterioles, the precapillary sphincters, and then the arterioles themselves. What do you think, arteriole? And basically how vasoconstriction or increased flow, whether or not that bypasses the capillary bed or whether that increases flow to the capillary bed. So certainly go over those as well as your Starling forces here. But we already described these two. Let's move on to venules and veins. First off, venules. And venules, not much to say about these at all. However, we do want to just remember that these have a thin wall, small amount of diffusion. Very similar to capillaries here for the venules. For veins, these are going to have a large internal diameter, large diameter. They're also going to have a valves, right? We talk about the one-way valves to keep flow going in the constant direction. They're going to have increased compliance, especially because of that very small, smooth muscle on their wall. They don't have as much force, so they can expand, they can accommodate more volume. This is why we refer to them as the volume reservoir. Okay. And then the Same sort of scenario that we described here as far as how we can compare the pressure change to the volume change. Here we're going to have a small pressure change for a large change in volume. Small pressure change or a large change in volume. Okay. And then some of the examples of the regulatory factors here was the venomuscular pump, right? So you should be familiar with the way that the valves, in conjunction with the skeletal muscle of the lower extremities, together helps to move blood in one direction back to the heart, so the muscular pump. You should also be familiar with the respiratory pump. This is how inhalation increases pressure in the thoracic cavity, decreases pressure in the abdominal cavity, creates a gradient for flow to go back towards in the large veins to go back towards the heart. And then finally, the venomotor tone, right? Venomotor tone. This is also in conjunction with the baroreceptors, the venous baroreceptors. But because we have that small amount of smooth muscle, when that smooth muscle exhibits tone, it actually helps to encourage blood to go back towards the heart. So this increases our um, end diastolic volume by increasing our venous return. That increases our ejection and therefore our mean arterial pressure on the ejection side, right, on the arterial side. So venomotor tone is one of the factors that increases mean arterial pressure on the arterial side. Lastly, the lymphatics. The function of lymphatics is to clear excess fluid. From the circulatory system. Okay. We also want to think about the volume. It clears about 3 liters per day, right? That's going from the circulatory into the lymphatics, being passed through the vessels and the nodes, then goes back into the venous system. So it's going to bring that fluid back to the venous system, right? And then some of the features that we find in our lymphatics as well is that there are valves in order to keep the direction in one flow or the flow in one direction. We have the thoracic duct as well. That's one of our major lymphatic structures. We kind of talked about this in lab yesterday as well. Major lymphatic structure draining most of the left side of the body and then all of your lower extremities, all of your abdomen and pelvis. And then we also talked about the role um, of our macrophages. Let me write this. The immune role of macrophages, and there's lots of macrophages in our lymphatic system, especially in our lymph nodes. Macrophages and lymph nodes.